ADHD, attention deficit hyperactivity disorder, which once again when I was at university, everyone knew was caused by eating red flavoured Smarties, red coloured Smarties. And they actually had this whole diet, I've forgotten the name of the diet now, that actually said if you've got a kid with ADHD, you know, they mustn't have raspberry balm, they'd have any red Smarties, had to take everything red out of their diet. Complete bunkum, absolutely nothing in it whatever. People started doing twin studies. It turns out ADHD is about 80% genetic, extremely genetic, identical twin correlation 0.8, non-identical twin half that. And once again, that's completely changed their gender. And my third example, multiple sclerosis, which people thought was just purely environmental, caused by a virus. People did a systematic study of all multiple sclerosis cases in Canada, asking if they were twins, and found this much higher concordance in identical twins than non-identical twins. Although those numbers look quite small, that's actually once again compatible with extremely high heritability for MS, and that once again has totally changed the agenda for how one goes about doing those studies. One could go on through the different realms of disease, even infectious disease, which you'd think, well, it's just whether you get the bug or not. But there are genes which actually predispose you to be susceptible to a virus or a bacterium or fungus or whatever, and there are genes which actually cause resistance. And you can see these are huge data in that the Scandinavians have these fantastic huge twin registries, because they can do record linkage of their health records with birth records and get complete data. And so these are based upon very, very large numbers. You can see there's something like 12,000 identical twin pairs and 20,000 DZ pairs, and finding these considerably higher correlations for infectious disease. And also even for cancers. Cancers tend not to have very high heritabilities, but there are genetic influences on quite a few cancers, particularly all the naughty bits, and also the lung. And lung is interesting because when people, now this is my only slide about molecular genetics, which is the other talk I could have given, but I'm not giving. This is the sort of light version. If we do genome-wide association studies, this is where we type genes right through the genome. We type about 500,000 gene variants across all 23 chromosomes, and then we actually do an association test of every one of those, what are called SNPs, single nucleotide polymorphisms, with our disease status, and see whether that p-value is remarkable, given that we've done 500,000 tests. And when you do that, in fact, you find this very significant hit on chromosome 15 for lung cancer, and guess what that gene turns out to be? It's a nicotinic acetylcholine receptor. And what does that tell you? This is actually a smoking gene. This is not a lung cancer gene. This is a smoking gene. This is a gene that actually predisposes you to smoking, and by predisposing you to smoking, it predisposes you to lung cancer. So is that a disease gene, or is that a behavioural gene? I'll leave you with that question. Interesting question. But incidentally, for those of you who think I only do twin studies, in fact, more than half my research is actually doing these genome-wide association studies of a whole range of different conditions. And this is the sort of result we come up with all the time, that you say, oh, such and such is a gene causing this disease, but actually what it does is predispose you to a behaviour which predisposes the disease. So another example is esophageal cancer. We know from epidemiology studies that the chief causes of, the chief risk factors for esophageal cancer that Christopher Hitchens died of recently are drinking and smoking. He was a huge drinker and a huge smoker. And the biggest risk factor, the biggest genome-wide association scan hits for esophageal cancer are the alcohol dehydrogenase genes and this nicotinic acid receptor gene, this same gene here. So there's just another example. Anyway, changing fields a little bit. So one of the things I study, because Queensland has the unfortunate distinction of being the world capital for melanoma, we look at melanoma risk factors. And the largest risk factor for melanoma is how many of these moles you've got, just common moles on your skin, because those are sort of like a pre-mutation, and it's well, at least half of melanomas are known to originate within existing moles. This is why there's such emphasis on these slap campaigns and things like that to try and reduce sun exposure. But we asked the question, well, sure, we know that sun is a risk factor because that's why Queensland has got such this terrible rate, but what about genetic factors? The genetic factors which predispose to moles. And so we've been counting moles on 12-year-old twins for about 20 years now, and these are our accumulated data on 422 pairs of identical twins and 762 pairs of non-identical twins, and you can see once again, hugely genetic how many of these moles you have. And this correlation of 0.94 between twin 1 and twin 2, incidentally, is about the same as the correlation you get as if you count the moles on the left-hand side and the right-hand side of your body. So that's just stochastic. It's just a little bit of developmental noise there that is lowering this below 1. So these are almost totally genetically determined. In fact, what is amazing is if you look at identical twins, you even see the placement of the moles on the body is in the same place. So how does that translate into risk for melanoma? So here's a pair of young Australian identical twins. These are diggers who were in the Pacific campaign in the Second World War. And those guys, when it got hot, they'd take their shirts off, they'd leave their hats off, they got incredible amounts of sun exposure, and their melanoma rates 50 years later are huge, and here they are 50 years later, and they've both got melanoma. And so we scoured Queensland and New South Wales for all melanoma cases who were twins. And although the numbers we got were not very large, we found 4 pairs of twins, identical twins who both had it, 23 pairs who were discordant, versus for non-identicals 3 pairs concordant but 95 discordant. And that actually translates into correlations of 0.55, 0.23. So over half the variation in melanoma risk is genetically based. And in fact, the rationale for doing this work and actually showing that is that it then gives you the stimulus to go on and actually try and find those genes using genome-wide association studies. So a large part of my work and the work of my group is actually in doing those population-based studies where we collect DNA from people and do genome-wide association studies trying to find genes for melanoma, of which we now have many. I've just sort of been very loose in my talk about how we separate different causes of variation, but being a bit more formal about it, we can actually break down the causes of variation in the population into 4 different components, 2 of which are environment and 2 of which are genetic. So the environment, we can actually, everyone talks about the environment, but you can actually split that into 2 categories. Those effects which you share with your siblings growing up in the same family because you live in the same household, you go to the same school, you share the same dietary habits or religion or whatever, versus those things which are unique to you and different from your brothers and sisters, unique environment. So those are the things that will make identical twin pairs different, whereas the C, the common or shared environment, is what makes twins the same. And then we also have 2 sources of genetic effects. Those which are additive, we just add up the effects of mum and dad, and that tells us what the child is going to be. But then those of you who've done genetics and know Mendel's laws will also know about dominance, that you can get one allele being dominant over another. So you actually have to have 2 of the same, you have to have the right combination of both alleles from your parents to see the same effects in a child and share that with a sibling. And so one can actually formally separate dominant effects from additive genetic effects. And so the twin model actually formalizes this by saying that what you have is a complete sharing of common environmental effects that correlate 1 between twin 1 and twin 2 in both identical and non-identical twins, whereas additive genetic effects are shared 1 between identical twins, but only half, due to Mendel's laws, between dizygotic twins. And by definition, unique environmental effects are unique to the 2 twins, and so they're not correlated. And so that enables you to write a pair of simultaneous equations that enable you to estimate these 3 quantities. And that's how I gave you a very crude example at the beginning, but in fact we're a little bit more sophisticated about it, and we can actually do weighted least squares, in which we weight these equations by the amount of information, the sample size, and so on, to get maximum likelihood estimates of these equations, of these quantities. So examples of this, this one might interest you, since most of you would have got here, because this is a Queensland core skills test that we have for all our 16-year-old twins. And this is on about 800 pairs of twins from Queensland, showing that performance in the QCST is about nearly 80% genetic, and only about 12% due to shared environment, family environment. So huge genetic influence on QCST. And just to be really subversive, we also measured IQ, verbal IQ, and performance IQ on the same people. And then we asked the question, well, how much of performance in QCST is due to the same genes that influence IQ? And the answer is all of it. It's, so this is a multivariate. So I just showed you the simple univariate model before. This is the multivariate model, where we expand that model to actually ask what are the causes of covariation between traits. Okay, why do we find that there's a correlation between QCST and IQ? And we can actually break that down into genetic and environmental effects. And what we see is that the correlation is that we have a single set of genes influencing verbal IQ, performance IQ, and QCST. We have some other genes that influence performance IQ differently from verbal IQ. Performance IQ is things like spatial ability, maths, digit span. It's sort of rather different from vocabulary skills and so on. And those, but then there is no extra genetic variance influencing QCST that is not already accounted for by these genes for IQ. Luke?
If there was no correlation or no link between the genes and the performance, what number would you expect for each of those?
If there was no relation between just the measured value of IQ and QCST.
Yeah, and just.
Well, if there was no, then all of these would be all of the cross paths all these diagonal lines would be 0 if there was no correlation. Yeah. So this, I mean, I'm giving this to you very quickly. I mean, the forerunner of this is the correlation between IQ and QCST is high. It's around 0.7, 0.8. And the question is that due to genetic reasons or environmental reasons? And are there any genes that are different for QCST than there are for IQ? And what this result is telling you is that all the genetic effects that influencing QCST are the same ones influencing IQ. That that QCST is not tapping any different genetic effects than you see in IQ. And in fact, the only thing that's different is this little bit of unique environmental variance there, which is about 10% of variance, and that's probably error of measurement. Anyway, I just thought, any education department employees in the audience? I just love stirring this up. Anyway, may as well give an IQ test. Do you remember all that agony you went through when you did QCST, all that stuff, you know? Just an IQ test, nothing else. So this